Hello, and welcome to Docera Clinical Education's Infectious Diseases podcast. I'm your host, Dr. Karen Straube-West. Today's episode features audio from a Medical Minute webcast and explores how recent clinical trial and real-world data can improve treatment plans for patients with PBC. In today's episode, I'm joined by Maria Londoño, MD, PhD. To view the full educational program and to download the accompanying slides, please visit the show notes for this episode. Now let's get started with Dr. Londoño.
Hello, my name is Maria Londoño. I am a hepatologist at Hospital Clinic in Barcelona, and today I will be sharing with you the latest development for the PBC management that were presented at the last EASL meeting in May here in Barcelona. So the presentation will be divided in three parts. First, we are going to discuss a little bit on the real-world evidence for the new PPAR agonist. In the second part, I will be discussing some of the data in patients who were previously treated with second-line therapies. And then at the end, I will comment on the patients who had an alkaline phosphatase between 1 and 1.67. So let's start with the first part, showing data on real-world evidence for the new PPAR agonist. So in this study, I will show you some data from the National Electronic Health Records in the US. This is a retrospective study analyzing patients with PBC with an alkaline phosphatase higher than 1.67 who received seladelpar after the approval by the FDA. The primary endpoint of this study was biochemical response defined by POISE criteria that you can see here in the slide. Means that the patient with response have an alkaline phosphatase lower than 1.67, the upper limit of normal, plus a reduction of more than 15% in the alkaline phosphatase as compared to baseline, and a total bilirubin lower than 1.1 time upper limit of normality. And the key secondary endpoints of this analysis was ILP normalization, and then at the end, they also wanted to check on side effects of the treatment. The cohort is divided in three groups. They are represented patients with PBC alone, patients with PBC and AIH variant that are so-called overlap patients, and then patients with PBC and MASLD. Almost 70% of the patients with PBC achieved biochemical response, 48% of patients with PBC-IH variant, and finally 64% of the patients with PBC and MASLD. In terms of ALP normalization, almost 50% of the patients with PBC achieved ALP normalization. And then at the end, they also wanted to show the data according to the cirrhosis status. Patients with cirrhosis had a significantly lower response rate as compared to patients without cirrhosis, 55 versus 68%. We also had data on elafibranor. These are the interim analysis of the Elfinity trial. This is a prospective phase 4 multicenter study in patients with PBC receiving elafibranor. In this first report, they show the data for the first 50 patients enrolled with month 3 outcomes. 54% of the patients already achieved biochemical response after 3 months of the therapy. And when they analyze the percentage of patients achieving ALP normalization in the whole cohort, 22% of these patients achieve ALP normalization. And when they analyze only patients with an alkaline phosphatase between 1 and 1.67, 40% of them already achieve ALP normalization after 3 months of the therapy. Both the whole cohort or patients who had previously received obeticholic acid, most of the patients present improvement or no change in alkaline phosphatase levels, and only a very small number of patients present worsening of ALP levels after the initiation of elafibranor. And finally, they also wanted to show us the data or the impact of the therapy on symptoms. More than 50% of the patients present an improvement on fatigue, and almost all patients present an improvement in pruritus, but this data need to be analyzed carefully because the number of patients with moderate to severe pruritus in this interim analysis is still very, very small. So in summary from this first part, we can say that in real-world practice, PPAR agonists were associated with an increased rate of ALP normalization and high rate of biochemical response. So the second part of this presentation will focus on patients who were previously treated with another second-line therapy, either obeticholic acid or bezafibrate. And these are the most difficult to treat populations. And we are very excited to see what is going to happen in the real-world data when we switch these patients from OCA or bezafibrate to the new PPAR agonist. So this first study is the real-world evidence from the Italian Early Access Program for elafibranor. This is a retrospective and prospective multicenter study in patients with PBC who discontinue obeticholic acid after the revocation of the commercialization in Europe. The primary endpoint of this analysis was the change in the median levels of alkaline phosphatase from baseline, and the key secondary endpoints were the proportion of patients achieving an alkaline phosphatase lower than 1.67, the upper limit of normal, or completely normalization of alkaline phosphatase levels. They also wanted to analyze the change in pruritus. There was a reduction in the median levels of alkaline phosphatase from 1.69 times the upper limit of normality to a median levels of 1.14 at the last elafibranor exam. And also, we can say that proportion of patients achieve levels of alkaline phosphatase lower than 1.67, 60% after 24 weeks of the therapy, and 1/3 of the study population achieved complete normalization of the alkaline phosphatase after 6 months on therapy with elafibranor. And also, in patients who had severe pruritus, there was a significant improvement in the symptom of at least 1 point, and the median improvement in pruritus score was 3.5 points. And then we present data from Spain, from the COLLIDE registry. Here, I will show you patients who receive seladelpar or elafibranor in real-world practice here in Spain. And the report is divided in different groups. Patients who are naive to second-line therapy, 136. Patients previously treated with obeticholic acid in combination with UDCA, 147. Patients previously treated with UDCA and bezafibrate, 82. And finally, a group of very difficult to treat patients who were receiving triple therapy with UDCA, obeticholic acid, and bezafibrate. For patients who are naive to second-line therapy, the reduction of alkaline phosphatase is between 30 to 40% already after 1 month of the therapy, which is very similar to what has been reported in the clinical trials. And the proportion of patients achieving biochemical response according to POISE criteria was 89% for patients with elafibranor and 68% for patients on seladelpar. But remember that the number of patients achieving or reaching this time point is still small. In the patients who had already experienced treatment with obeticholic acid, the reduction of alkaline phosphatase after 1 month of the therapy is around 20 to 30%, which is a little bit less than for naive patients. But again, the proportion of patients reaching a biochemical response after 6 months of the therapy is very high, more than 80%. And when we analyze patients who were receiving UDCA plus bezafibrate, we can see that between 40 to 50% of the patients achieve biochemical response after 6 months of the therapy. And finally, in patients who were previously treated with triple therapy, UDCA, obeticholic acid, and bezafibrate, the results are very different as compared to the previous groups. Instead of a decrease in alkaline phosphatase levels, you can see an increase in the levels of alkaline phosphatase in both drugs, elafibranor and seladelpar. And then at the end, at 6 months, 68% of patients remain on response according to POISE criteria for elafibranor, and 49% of patients receiving seladelpar remain on biochemical response according to POISE. And finally, it's important to mention that a significant proportion of patients here, 23% and 15%, lost the response according to Paris 2 or POISE criteria after discontinuation of the triple therapy. So in summary from this part of the presentation, we can say that after switching to elafibranor or seladelpar, we can say that the response rate in naive patients is still very high and similar to that reported for the clinical trials. And in patients who were previously treated with obeticholic acid or bezafibrate, the response rate is still very good. But the problem is for patients who were receiving triple therapy, in which there was some loss of response after the discontinuation of the obeticholic acid and bezafibrate. So for the last part of the presentation, I will be focusing on patients who have alkaline phosphatase between 1 and 1.67. So this is an important population because in the clinical trials that led to approval of both elafibranor or seladelpar, they were only including patients with an alkaline phosphatase higher than 1.67. And now we are providing new data showing that the response in patients with mildly elevated alkaline phosphatase are very, very good. So let's move now to the first study. So the first is it's a real-world study in patients receiving elafibranor. So the data here is extracted again from electronic health record data in the US called Health Verity, and the analysis focused only in patients who had alkaline phosphatase between 1 and 1.67. And the data is divided in two groups, in patients who were naive to second-line therapy and in patients who received another second-line therapy previous to the initiation of elafibranor. In both groups, previously treated or naive patients, there was a significant decrease in ALP levels after 6 months of the therapy, with no significant changes in bilirubin levels. And in terms of response, you can see here that patients naive to second-line therapy, 60% of them achieve complete ALP normalization after 6 months of the therapy, and 46% of patients who were previously experienced or who had previously been treated with a second-line therapy achieve complete normalization of alkaline phosphatase. And finally, we have some data from the ASSURE trial. Just to remind you, the ASSURE trial is an open-label extension of the RESPONSE trial, which is the phase 3 clinical trial that led to the approval of seladelpar. And in this open-label extension, patients in the RESPONSE trial can start this open-label phase. And also, there were patients from other seladelpar legacy studies that were able to participate in the ASSURE trial. And this specific analysis focused again in patients with mildly elevated levels of alkaline phosphatase between 1 and 1.67. A significant proportion of patients achieve complete normalization of alkaline phosphatase, which is 76% after 24 months of the therapy, and a significant number of patients achieve a more than 15% reduction in alkaline phosphatase, again at 24 months of the therapy, 85%. And then again, from the ASSURE trial, there are important data analyzing the impact of response on liver stiffness measurement. Patients who achieve complete biochemical response defined by Boyce criteria, the liver stiffness measurement is stabilizing or even improving a little bit after 36 months of the therapy. And on the contrary, patients who did not achieve biochemical response present an increase in liver stiffness measurement during the follow-up. They did some univariate analysis and found that old age, low levels of alkaline phosphatase, and complete biochemical response were associated with improvement in liver stiffness measurement. And in the multivariate analysis, the only variable that was independently associated with changes in FibroScan was the achievement of biochemical response, and then the impact of biochemical response in the changes on FibroScan. The odds ratio favors all the time biochemical response. So patients who achieve biochemical response, either complete normalization of alkaline phosphatase or biochemical response according to Paris criteria, show an improvement or a stabilization of liver stiffness measurement during the follow-up. And then in summary from this last part of the presentation, we can say that in patients with mildly elevated alkaline phosphatase between 1 and 1.67, the upper limit of normality, PPAR agonists were associated with high rates of biochemical response and ALP normalization, and in the ASSURE trial, biochemical response is also associated with stabilization or improvement in liver stiffness measurement. Thank you very much for joining this program today. and I hope you like it. Thank you.
Thank you, Maria, and many thanks to you, our listeners, for joining us. To view the full program and download the accompanying slides, please click the link in the show notes. To get access to all of our new podcasts, subscribe to the DoCERA Clinical Education Podcast on Apple Podcasts, Google Podcasts, or Spotify. Thank you, and have a great day.